Thank you for your interest and for attending this short presentation. My name is Katja Manninen, and I'm a field application scientist at Invivoscribe. I will talk to you about leveraging highly sensitive next-generation sequencing assays for clonality, somatic hypermutation, and MRD assessment. Here is the agenda for today's talk. I will start with the brief company introduction. Then I will present clonality detection by next-generation sequencing, our LymphoTrack assays and software, as well as the next-generation sequencing workflow steps. We will end the presentation with somatic hypermutation and MRD detection. Let's start with the brief overview of the company. Invivoscribe currently has 6 companies in 5 countries, as shown here on this map. Our research and development, quality, regulatory, data analytics, and our FDA-registered cGMP manufacturing teams are all located in the US. We have clinical reference laboratories, distribution hubs, and offer technical support at our companies in the US, Europe, Japan, and China. We have over 700 customers, more than 75 pharmaceutical partners, and we provide our products and services to more than 160 countries. Throughout our almost 3-decade-long history, Invivoscribe has focused on standardization of our assays and our ability to provide these assays to the international community. Invivoscribe provides a full range of standardized CE-marked in vitro diagnostic cGMP products for hemato oncology, as well as RUO assays, analyte-specific reagents, and DNA and RNA controls. As a premier diagnostics products manufacturer, we only offer superior quality products. Our experienced regulatory team has over 50 product registrations and multiple companion diagnostics approvals in the US, EU, and Japan. LymphoTrack assays are next-generation sequencing-based clonality assays that follow one single protocol for all available targets and platforms. The assays can be used for clonality assessment, somatic hypermutation detection, as well as MRD detection. The LymphoTrack kits are a complete solution. The kits contain ready-to-use indexed PCR master mixes, the necessary controls, and they come with bioinformatics software to analyze your data. The primers are designed with barcoded indices and adapters, so the sequencing libraries can be generated with one single PCR, which streamlines the overall workflow and eliminates the need for post-PCR ligation step, and therefore reduces the potential for sample cross-contamination. The LymphoTrack assays identify and assess the mutation status of B and T cell gene rearrangements. The targets are shown in the center in the light blue circle. The available B cell targets are IGHV leader, framework 1, framework 2, and framework 3, as well as IG kappa. The T cell targets are TCR gamma and TCR beta. The workflow consists of one-step PCR for amplicon and library generation. All the assays are compatible with each other, and you can pool and sequence amplicons from any LymphoTrack kit together, which reduces the per sample cost of testing. Bioinformatics software is available in desktop or Docker images for analysis and interpretation. The bioinformatics software is able to sort the complex NGS data for multiple targets for easy analysis and visualization of individual samples. The same LymphoTrack kit reagents are used for clonality, somatic hypermutation testing, as well as MRD testing. All LymphoTrack assays follow the same 7-step procedure shown here. First, you will extract DNA with the method validated in your lab. Then you will set up one-step PCR for target amplification with the indexed PCR master mixes. After PCR amplification, we purify the amplicons with AmpureX bead cleanup. Then the amplicons are quantified with kappa, qPCR, or tape station or bioanalyzer and pooled and loaded onto your DNA sequencer. After the sequencing run is complete, we analyze the FASTQ files with the LymphoTrack software. The multiplexed assays can be run and set up side by side to reduce laboratory time, and the MySeq reagent kit is selected based on the longest target amplicon you are sequencing. This very simple hands-on workflow ensures that even the new NGS users can succeed with ease. LymphoTrack kits and software are supported on both Illumina and Thermo Fisher sequencing platforms. Data analysis of the FASTQ files is done with the LymphoTrack software. The desktop application is intended for low and medium throughput labs, and the LymphoTrack software package is provided with each LymphoTrack assay kit. The software is composed of 3 distinct parts, which are the LymphoTrack data analysis application, a LymphoTrack reporter plugin that runs automatically after analysis, and an optional LymphoTrack visualization method that consists of 4 Microsoft Excel spreadsheets. The software runs on supported Microsoft Windows platforms. Using the software is done in 4 simple steps shown here. Number 1, select the targets you would like to analyze by checking the appropriate checkboxes. This can be just 1 target or up to all 7 targets. Step 2, choose your decimal mark that you would like the data in your reports to be displayed in. Step 3, browse to the folder where your FASTQ files are stored at. And in step number 4, you will launch the analysis. The analysis should take around 5 minutes per sample per target, and the output is in the form of PDF reports. Besides the LymphoTrack desktop software, we offer LymphoTrack Enterprise software. This is designed for higher throughput laboratories that generate high volumes of sequencing data using the LymphoTrack assays and LymphoTrack MRD solutions. When used with the comprehensive LymphoTrack solutions, the enterprise software enables a fully automated pipeline for bioinformatics data analysis using the raw FASTQ files that are generated from Illumina MySeq, NovaSeq, and NextSeq instruments. This Linux-based software is built for portability, flexibility, and can easily be integrated into downstream reporting systems and your LIMS system. Continuing with Invivoscribe kits. Somatic hypermutation status provides an important prognostic indicator for chronic lymphocytic leukemia, which is a very common type of mature B-cell leukemia. Somatic hypermutation testing is performed as a standard of care in CLL. Invivoscribe offers solutions for somatic hypermutation assessment with both LymphoTrack IGHV Leader and Framework 1 assays. The same LymphoTrack NGS kits and LymphoTrack software that are used for clonality testing can be used for somatic hypermutation assessment. One key benefit is that NGS assays are easy, less time-consuming, and more sensitive than your traditional Sanger sequencing method of somatic hypermutation testing. The LymphoTrack assays can also be used for MRD detection. We offer a comprehensive solution for MRD clonality testing, and this bundled solution consists of the LymphoTrack assays. These are the same assays that are used for clonality detection of the initial sample. It also includes B and T cell controls, and these controls are the LymphoTrack low positive B and T cell controls, as well as the LymphoQuant internal control that is spiked into the positive control and your samples. The LymphoTrack MRD software for seamless sample analysis completes this bundled solution. Some of the advantages of the MRD solution include the ability to offer concordant, objective, and standardized testing worldwide by tracking sequence-specific DNA targets. You have the ability to detect clones, newly emerging clones, and subclones in follow-up samples, which allows you to see how well cancer has responded to treatment, and you can confirm and monitor remissions. The level of sensitivity and confidence of the assay is only limited by the DNA input amount tested and the sequence read counts that are generated during the sequencing run. You can consistently assess and track up to 5 clonal sequences over time. The LymphoTrack MRD software is designed to work with the MRD controls to automate MRD analysis of up to 5 clonal sequences simultaneously. MRD software generates PDF reports which provide estimates of clonal cell equivalents and graphs to visualize changes in clonal frequencies over time when you track the sample over multiple time points. A very important step in MRD analysis is to first plan the level of confidence you want to reach, meaning how sure are you that an MRD negative result is truly negative. And the MRD software has an experiment planner tool for this. The addition of a spiking LymphoQuant internal control to your positive control and samples allows the software to convert percent clonal reads into estimated clonal cell equivalents. Also, the addition of the LymphoQuant internal control facilitates clonal tracking over time without additional sequencing run cost. Consistent use of the LymphoQuant internal control enables clinicians to objectively monitor the disease over time with the highly standardized and sensitive method. We also offer FLT3 ITD MRD and NPM1 MRD assays. These targeted deep sequencing assays can easily be implemented together because they follow identical workflows. They are designed for high-throughput laboratories, and each kit configuration includes 24 dual-indexed PCR master mixes to help prevent cross-contamination due to barcode hopping. Dockerized FLT3 ITD MRD software and NPM1 MRD software seamlessly integrate into downstream reporting systems and LIMS and simplify interpretation by providing objective results in a TSV output file. Some of the key benefits of these two assays include allelic sensitivity level of 5 times 10 to the minus 5. Baseline sample is not required and you can test your sample at any point in time. You are able to bring standardized FLT3 ITD MRD and NPM1 MRD testing in-house for faster turnaround time. You can batch samples using previously isolated genomic DNA, and you have the flexibility to multiplex targets and up to 48 total samples per sequencing run for increased cost efficiency. FLT3 ITD MRD and NPM1 MRD assays have real-world impact, as demonstrated in the study that tested the hypothesis that detection of specific residual AML-associated variants in the blood of patients in first remission prior to allogeneic hematopoietic cell transplant would be associated with higher rates of relapse and mortality after transplant. The study showed that among AML subjects in first remission, the presence of FLT3 ITD or NPM1 mutation in blood at an allele fraction of greater or equal than 0.1% was associated with increased risk of relapse and worse survival compared to other AML-associated mutations. This study provides strong evidence that using next-generation sequencing as the diagnostic platform for MRD testing of subjects with either FLT3 ITD or NPM1 mutated AML in first remission prior to transplantation could identify differential risk. I would like to end with the summary of the Invivoscribe ecosystem and emphasize that precision diagnostics is the foundation for precision medicine. Our LymphoTrack and LymphoTrack MRD assays, together with our software, identify and track cancers at the molecular level for both B and T cell targets which enables proper diagnosis and a way to track your patient's response over time. A clinical test is the most effective only when the test, interpretation of the test, and the results reporting are internationally standardized, just like Invivoscribe has done across our multiple international locations and laboratories, and with our cGMP manufacturing of reagents and kits. For nearly 30 years, Invivoscribe has developed, manufactured, and commercialized the gold standard molecular hematopathology assays and reagents for gel and capillary electrophoresis detection, and most recently for next-generation sequencing detection. Thank you so much for attending this short presentation. Please scan the QR codes for more information on Invivoscribe products and services.